Hello, everyone. Thanks for joining us today. I'm Charles, and I'm the CEO of Satena Bioprocess Solutions. Today is my honor to represent CELLINK Group, and also I will present our novel technology and products, also how we are going to revolutionize the future of the medicine. Before we start, I would like to give a short introduction of CELLINK Group. We are a world-leading bioconvergence company and focus on cutting-edge technology, products, and service to our global customers, including biopharmaceuticals companies and the top research institutes. It basically includes about creation, about understanding, about master biology. And our goal is creating a future of medicine together with our customer to develop game-changing solutions by combining biology and technology. And let me start with the latest overview of our product portfolio, since this nicely shows where we are doing at CELLINK and where we are coming from. Our product cover a broad range from single-cell isolation, single-cell omics, and microbioreactor liquid handling system, also dispensing live cell imager, and also 3D cell printing. I will walk through the technology in my following presentation today. First is about the 3D bioprinting, and it's basically where we start. There are several challenges in current medicine. So first of all, in average, a medicine from lab to shelf takes more than 10 years of research in average. And the second, more than 20 people dies every day just because the lack of the organ transplants. And also researchers still use mammalian models for drug testing, which well known not an ideal model. And it can waste up to 2 million every year. So the 3D bioprinting technology potential can solve all these issues. First example is as far as scaffold-free approaches are concerned, the most common applications are for spheroid and organoid development. There are small bunches of cells from a specific tissue that self-assemble into spheres and start to mimic the tissues they're coming from. Researchers have come up with many different ways to create these spheroid, as you can see here. This is especially beneficial for cancer research. For example, if you think about a tumor, in essence, it is a ball of cells that grows uncontrollably. But in 2D, you are not able to mimic geometry. This often leads to an inaccurate result for drug discovery research, which costs pharmaceuticals companies both time and money. Creating a tumor model in 3D allows the cells to behave as they would in the body, making drug testing more accurate. The same is true for scaffold-based approaches. When cells are mixed with the materials that make it easier for them to self-assemble, researchers can create more functional models faster. And when these cell-laden materials are 3D printed into a correct geometry, the researcher can leverage the combined benefits of the biomaterials and the architecture to create a model that more accurately mimics that in vivo environment. And also another example is from our collaborator CTI Biotech, who has focused their 3D bioprinting skin tissue model on in vitro testing of cosmetic products. The skincare business is a huge market that long has been depending on animal model for testing, which is ethically unsustainable. And a lot of effort is being focused on the establishing in vitro models that directly can replace the traditional animal models. There are already commercially options available, but to advance and specialize these models to get a more complete picture is an ongoing work, and CTI has kindly provided us with some images of their skin model shown here. And with the incorporation of the model in the skin tissue models, where is the possibilities to model the oiliness of skin, which varies a lot between individuals and affects which skincare products that are more suitable. And also in general, skin models like this allows for testing, both topologically and as well as injection tests, where more relevant and accurate results are the outcome. And other than 3D printing technology, let's talk about application of our bioscience group a little bit. We offer our products to a wide range of life science customers in many applications, and I will walk you through some specific workflow in a minute. In general, you can summarize our application area into biopharma cell line development for manufacturing antibodies, or cell and gene therapies, liquid handling, where we have unique technology for volume reduction, and live cell imaging with some great microscopes and analysis software. And we offer some interesting products and consumables, especially for sample preparation in single-cell analysis or omics. The first workflow I would like to speak about today is cell line development. Cell line development is a key process in bringing biopharmaceutical drugs to the market. Most best-selling drugs today are produced by cell lines. The cell lines need to be derived from a single cell as required by the FDA. So traditionally, cell line development is a very complex and expensive process, and it is on the critical path toward market launch. That means every week you save lets you bring your drugs to the market earlier. And remember, there are many multi-million dollars biopharma drugs out there that might only have 10 years or so left under patent protection when they finally hit the market. A week of saving is very substantial. Now, when you look at our workflow at the bottom of the slide, you will see the upside. The upside combines multiple instruments in one, and combined with SNES, it saved up to 13 weeks in the cell line development workflows. Assuming a pharma company make $1 billion annually with a drug under patent protection, 13 weeks more offers a potential for more than $250 million in revenue before competition join the market. And we also have a beautiful workflow in next generation sequencing. The go-to method study genes. This workflow can be used for single-cell sequencing, but it's also helping around the world to ramp up sequencing capability for finding COVID variants. In the case of a single-cell sequencing, the user starts by sorting a single cell with one of our single-cell sorters. The full workflow includes our single-cell sorters like offsite and also seq-well reagents and the IDOT dispenser, and also the C-watch for DNA purification before the sample go into the sequencer. And when you study effect of new drugs, you typically test them on cells. To do this in a consistent and high throughput manner, we offer a simple yet powerful workflow. You start by treating your cell, wash them very consistently with a C-watch, and use the cell side X for time-lapse live cell imaging. The result can be analyzed in our Cell Site Studio software. It's very easy of use. This is something that is very close to our hearts. And here I would like to also introduce a little bit more about high IDOT technology work. The system uses up to 8 individual controlled positive pressure channels to generate droplets from 8 to 15 nanoliters from a small hole at the bottom of each well. Each channel can generate up to 400 droplets per second. IDOT's drop detection is a patented feature and detects and counts every droplet released during a single dispensing run. It's a simple and powerful tool enabling droplet verification and protocol optimization. Drop detection is a circuit board mounted under the IDOT source that leverage 96 miniaturized light barriers to detect every droplet generated from each source plate position, identifying change in light intensity to detect droplets as they pass the light barrier. After dispensing, drop detection produces color-coded and text file-based results. And for the other products, I just want to use a video to walk you through this technology I just mentioned a few minutes ago, including S-Nest, Upside, and also Cell-Side X. I believe you will have better understanding after seeing this video. Okay, we also have a slogan in the groups, always the customer's best at heart. We believe our customers are like our family, so we do everything we can to help our customer being successful. So please come reaching us. I believe you will find lots of amazing products and technology, and also understand how we can help with your research and workflow. Thank you very much.